Good morning, everyone. My name is Will Simpson. I'm the Director of Communication for PolarisQB, and we're very excited to be sharing with you today our webinar on quantum-aided drug design and delivering business value through Quad. Our speaker today will be our CTO, Bill Shipman. Bill Shipman and our CEO, Shahar Keenan, founded the company in Durham, North Carolina in 2020, and he will be telling us all about how we are using quantum utility today to provide value to customers and partners, and what the quantum-aided drug design system is able to do. So, Bill, I'll hand it off to you.
Very good. Thanks so much, Will. Really appreciate the warm introduction. So again, I'm Bill Shipman, CTO and co-founder of Polaris Quantum Biotech. I'm really excited to describe how Polaris is providing business value with Quad to our customers and partners. All right, so first let's start off with our mission statement. It's a pretty bold mission statement. We're designing drugs to treat and cure all diseases for all people. That is a very ambitious goal, but it's why we get up and come to work every day and push forward with our mission. So as Will said, we were founded in 2020 in February in Durham, and we've grown up a team of 10 now. We're very excited and very proud of the accomplishments. Very notably, we have the first drug discovery platform leveraging the power of quantum annealers. And late last year, late 2023, we launched Quad, the first quantum SaaS product available for the drug discovery market, which there's a lot of hype about what quantum computers can do in the drug discovery market. Here's the first SaaS offering in that space, which we're very proud of. So we're talking about drug discovery. Let's place Polaris in the spectrum of what the drug discovery process looks like, and let's highlight a couple key metrics from this visualization. So we'll see that from this visualization that the time period for drug discovery is on the average a decade to 15 years if you sum up the number of years spent in pre-discovery and clinical trials and FDA review and then approval. On average, that's a decade to 15 years of time. Polaris is tightly focused in the drug discovery phase of time, so that first 3 to 6 years, we can step forward into the preclinical space for the very beginning of it, so early stage assays, but we are primarily focused in the design and discovery of novel drugs. So let's talk a little bit greater depth on what drug discovery is. So fundamentally, it's a multi-object optimization problem. I think we can all appreciate that it's long, as we've just seen in that prior slide, as well as from the media. We're talking about years of effort with very uncertain outcomes, so significant time reward risk there. Very expensive process. Again, as I think we know from the media, we're talking about billions of dollars of investment to get to an approved drug. Significant amount of that expense is on the back end where we're not operating in clinical trials, but there is a non-zero number that we're going to look at in another slide, talking about how much cost and time is spent just in the discovery phase. That's dominated very much by wet lab costs, so doing synthesis and testing, and then other processes associated with that. One feature that is not well characterized in these media narratives and what we conventionally see is the limitations of current processes. So they're dominated either by high throughput screening or by very traditional CAD approaches, which those are looking at hundreds of thousands to millions of molecules, a very small slice of total chemical space that is relevant for drug discovery. So to expand upon the current industry process, it's essentially a set of sequential optimizations. So the very first place we have to start is binding a small molecule to a protein of interest. So that would be the first optimization you should undertake. That process is going to be doing probably some CAD work, computer-aided drug discovery or design, doing synthesis, testing it, analyzing results. If we're not coming up with the right binding affinity we need, we get to sit there and loop until we found success. Then we can transition to the next sequential optimization, in this case solubility, although it could be others, followed by a third optimization and then a fourth, each time likely doing some computational work doing synthesis, doing testing, analyzing results, iterating until we find success. On average, 3 years and $4 million to get to lead-like candidates. That's a significant investment of time and capital for uncertain outcomes. Polaris changes that paradigm using quantum annealers, and instead of doing sequential optimizations over multiple years, instead is phrasing the problem as a single optimization problem on a quantum annealer, and is able to look at large chemical spaces, billions of molecules, to find the best lead-like molecular candidates from the chosen search space. Again, that's a very rapid process for us. We'll talk about that in a little greater detail in just a minute. So, what is quad? Quad fundamentally takes as input a small protein structure, 3D representation. That's shown here in green. The binding pocket of interest is highlighted in silver. From the binding pocket, we then calculate the appropriate chemical space to use. We can either draw fragments from de novo chemical space, so novel synthesis. We can also draw fragments from on-demand libraries, so Wuxi, Anamine, Ottava, LCC, depends on the nature of the project and the interests of the partner who they prefer to work with or what synthesis they want to undertake. Having the 3D structure and the fragment library, we then can express that in the form of a QUBO, quadratic unconstrained binary optimization, it's quite a mouthful, to D-Wave's annealing system, which will then optimize the fragments and generate a library of lead-like molecules, roughly 3,000. This entire process takes 30 minutes. If we factor in some time for busyness of the annealer, again, we're using it as a cloud service, maybe hours, but very tractable amount of time, order of days. In principle, this part of the process takes a half hour. It's a significant time savings to get to a library of 3,000 lead-like molecules. So we've talked about quantum annealers. Let's take a step back and for just a minute and talk about quantum computers writ large, since they are a very popular topic of significant conversation. So we can divide the world into two primary modalities of quantum computers. On the one hand, you have quantum gate computers. Those are very interesting, and there's a lot of promise in the future there. We can conceptualize them in the way that our current computers that we're watching this Zoom call right now operate. It's manipulating a qubit, which can have so-called discrete states in the way a digital computer does, ones and zeros. It's just you get more states per qubit on a quantum gate computer. That being said, the largest ones in commercial production right now are order of 100 to maybe 1,000 qubits. The only one I know we've touched is on the order of 120 qubits. And those get to solve very general purpose problems. Looks much more like a general purpose computer. Quantum annealing, on the other hand, operates as an annealing system going from a high energy state to a low energy state. As long as you can phrase your problem as an optimization problem and can traverse that energy gradient, a quantum annealer can solve your problem. Not all problems can be phrased as optimization problems, but many problems that you wouldn't recognize as an optimization problem can be reformulated that way. With D-Wave's quantum annealer, we've got roughly 5,000, 5,500 qubits to work with. That's an order of magnitude greater than what's available for gate systems, and it's just a more robust system for the kinds of problems we are trying to solve. To look at a couple other names we might recognize from the industry. So certainly we've highlighted D-Wave. That's who we're working with primarily today. IBM has a processor, I believe it's called the Eagle processor, which is what we used on a DARPA contract that's got order of 120 qubits. Very impressive piece of machinery, but it does not have the expansive capabilities that we need to solve our optimization problems today. There are two other notable players in the industry, IonQ and QEra. They have very distinct technology paths. I'm a software engineer. I can only speak to a limited degree about hardware, but very excited about what they're doing. They are proposing to have 1,000 and 10,000 qubit systems online in the future. I'm not sure what that roadmap is. There's a lot of potential here. There is value to unlock in quantum computing. Right now, we are getting utility from D-Wave today. That is where the industry is, and that's how we are deriving value from quantum computing today. So, to step back again, why quantum and why now? Again, we're talking about speed and significant cost savings compared to conventional approaches, whereas conventional approaches are talking years and millions of dollars. We're talking about getting to lead-like libraries in the order of minutes, hours, days. The size of the total permutable space we're looking at is 10 to the 30th molecules. We certainly are not looking at the permutations of 10 to the 30th on 5,000 qubits on D-Wave. We are looking at the subset of 10 to the 30th that is relevant and appropriate for any particular project. Whether it's a kinase or a protein-protein interaction, we are constraining the chemical space to what is appropriate for that particular project. Finally, I'd highlight here the ability to rapidly go through iterative cycles as is appropriate for a project, whether the iteration might be testing competing hypotheses for binding modality, or perhaps wanting to explore more chemical space by substituting fragments in, or perhaps looking deeper into a particular bit of chemical space by, again, changing the fragments or optimization parameters. That is not as reasonably tractable in traditional approaches, given the time considerations of traditional approaches. So let's get into the meat and potatoes of why I think everyone is probably here, and that's to talk about case studies. So the first case study that I want to highlight is comparing quad-produced molecules to more traditionally created molecules. So again, going back to the initial process of quad, we need to start with a binding pocket of a protein and a template molecule. Template molecule is shown in left in green. We see a quad produced molecule in right in red. You'll see that the quad produced molecule is occupying very similar space in the binding pocket. The protein here is NNMT, and the ribbons in yellow and brown are the structure, the three-dimensional configuration of the protein and binding pocket. Seeing that we have a small molecule produced by quad fitting into a similar manner as the template, we can highlight different features that the quad produced molecule has relative to the template. So the template molecule with the fused ring system on the left extreme is sort of in a more horizontal orientation, whereas the quad produced molecules are perhaps in a more vertical orientation. Those are actually different fused ring systems as well. This is indicating quad is able to explore the binding pocket and come up with other reasonable molecules. This may affect solubility or binding affinity, variety of different parameters that this can have an impact on. So let's compare quad performance to a larger set. Instead of a one-to-one comparison of the template to the quad produced molecule, let's instead consider quad molecules compared to a library produced with traditional CAD approaches. So in both these graphs, we're looking at quad produced molecules that are roughly 2,000 molecules in the library, and the CAD library had 2,800 molecules. So in a direct heads-up comparison at the same strength of binding affinity, the quad library had 323 molecules that had high level of high binding affinity versus traditional CAD approach with 11 molecules. If we rescale that, we can consider that as an enrichment factor, and the quad produced molecules had an enrichment factor of 42 as compared to the traditional CAD approach. Again, this is work that is done in a matter of minutes, hours, rather than weeks, months, or years. Another way that we can consider quad produced molecules is to compare them to known binders. So this set of binders, roughly 60 binders pulled from Kimball for NNMT. We compared the TPSA of the known binders from Kimball to quad, as well as the number of rotatable bonds for the Kimball-produced molecules to the molecules found in quad. We'll see that the Kimball molecules have a much larger range of values because while they are good binders, that does not necessarily mean they are drugs or have drug-like properties. Whereas the 2000 molecules found by quad all exist within the narrow range of appropriate values for TPSA and number of rotatable bonds for drug-like molecules. So we've now looked at comparing a quad-produced molecule to the template molecule. We've compared quad molecules to molecules found with traditional CAD approaches, and we've looked at quad molecules relative to other known binders in the public domain. Before we transition to the next case study, I do want to pause here and highlight a couple different operating modes of quad. So there is a best-in-class mode that prioritizes similarity of the library to the template structure. So that would imply that we're looking at a very narrow portion of chemical space relative to the template structure. So we're able to look and get greater resolution on that chemical space. Quad can operate in a first-in-class mode. So that again is looking at the template or a perhaps homology model to define what is the appropriate binding pocket we want to use, characterize that, and build a fragment library that instead of staying very close to the template, instead looks at a very large chemical space, may look at very discrete portions of chemical space to find appropriate drug-like molecules, lead-like molecules. Another modality would be scaffold hopping. That's where we take, or the user, whether it is Polaris operating quad or our partner operating quad, takes the template structure and identifies key regions that they want to maintain or swap out, whether that's perhaps a tail of the molecule or the central scaffold, depending on the parameters of the project. Currently, quad is able to work with small molecule templates, peptides, and macrocycles. With that, let's step ahead to look at our second case study. So this is specifically looking at quad's best-in-class mode. The template here was sorafenib, looking at the protein P38 MAP kinase, a very important oncology target. On the right-hand side in blue, we see the three-dimensional rendering of P38 MAP kinase. We see sorafenib laying within the binding pocket. The left-hand side, we see the suggested fragmentation scheme. So this is a screenshot from quad highlighting the scissable bonds that sorafenib can be broken up to express either the fragmentation schemes that you're interested in. Do you want lots of small fragments, several large fragments, or alternatively, this same interface is used for scaffold hopping. So if you want to save the left extrema, you would annotate the scissable bond you want to break and mark that fragment as held constant. Sort of an overview of how quad works in best-in-class mode. Let's look at the results that came out of running best-in-class on sorafenib. So it produced roughly 3,300 molecules in the library. There was a lot of novelty in those molecules. We could readily find 3 different molecule series that were already known that were not covered by the sorafenib patent. Some of these molecules are simply described in literature. Some of them are in other patents. So what's the value proposition here? You might ask, well, you can engage in perhaps patent breaking, or you might use the tool to expand your patent so that you can find other relevant structures that are non-obvious to ensure those are covered in your Marcouche set, so you don't immediately have a fast follower in easy access relative to your covered IP. If we look at a heat map, looking at the entire library that was produced in this work of the roughly 3,300 compounds, this heat map is created using max keys for the molecules. So that's essentially an identity function to be able to relate the similarity of one molecule to another. So looking at this heat map, you see that there's significant diversity, but nice areas of clustered molecules that have a higher degree of similarity to each other relative to the rest of the set. The left visualization is what you might conceptually see if you were to look at very fine resolution, actually look at visualization of those molecules. You'll see the right side of the visualization, there's a 6-member ring that's more or less constant in that binding pocket. There is some flutter about it, but it essentially sits there. And I would imagine that's part of the reason it's clustered in that way, whereas there's more variation on the left-hand side. So this is sort of a summary of how quad works for best-in-class mode. We're finding both novel molecules in reasonable chemical space, as well as identifying other known examples of prior art and binders. To summarize some of the high points of quad, we have worked with in 2023 kinases, PPIs, and transmembrane proteins, amongst others. The results we've been getting from the libraries and from the downstream processes match well with the prior data our partners and customers have. So we are seeing both repeat customers and customers who are finding a high degree of satisfaction with the molecules we are producing, both because they are meeting the target activity as well as they align to prior data. We've had a couple customers who had lack of success with other partnering activities, so very gratified to see that. We are able to rediscover known drugs, as was shown in the sorafenib case study, as well as we are finding more drug-like molecules than binders found in Kemble. To bring the presentation more to a close, I'd like to highlight a couple key takeaways that I'd like you all to leave with. Polaris is looking at vast chemical space. We have repeat customers. On the quantum computing front, I would highlight that there's a lot of hype around the concepts of quantum advantage, quantum supremacy. In drug discovery, I wouldn't say those are really relevant or germane. We need quantum utility. We need to be able to use a new tool to drive value and meaning from it. We are doing just that with quantum annealers today. We're looking at incredibly large chemical spaces and generating drug-like molecules for downstream processes. That is the definition of utility. Finally, we have multiple paths to work with partners. We welcome conversations to find how we may assist you in finding your star molecule. And with that, I'll pass the baton back to Will so that he can lead us in the Q&A. Thank you very much.
Thank you, Bill. We hope you've all enjoyed seeing a little bit of how we're using quad and quantum utility today to drive real value for our customers. We're very excited about the quad system, and as Bill has said, we've had a number of customers that have gotten very good results and have been very happy with what they have been able to drive in a much shorter time frame than previous processes. So thank you all for attending today, and we look forward to hopefully seeing you in the very near future.